Hey everyone, welcome back to DIY Biotech. Today I have a very exciting article that was published this month, end of last month, really recently from the Jones Lab on producing really high quantities of psilocybin in genetically modified bacteria. So this is the paper that we will be looking at today. It's in the journal Metabolic Engineering, as you can see, which, as I've discussed in previous videos, is a very, very good journal for this field. This is what I did my PhD work in, was in metabolic engineering, and I have my own paper published in this journal. It's a great journal. Jay Kiesling is one of the editors, a professor at UC Berkeley. You can look him up. Also a fantastic research lab. But this paper comes out of the J. Andrew Jones group at Miami University in the great state of Ohio. Yeah, that's right. A little bit confusing, but this is a lab that is mostly ran by master's students and undergraduate students, which is really cool. If you want to see a sit-down conversation with myself and Dr. Jones, that was previously published on this channel. Really enlightening. He's a really great PI and does really interesting work in producing psychedelics or these tryptamine compounds in bacteria. So let's begin here. Previously, the last paper that we looked at from the Jones group that produced psilocybin in bacteria, they were making this quantity 1.16 grams per liter of psilocybin in bacteria, which is already a huge quantity, right? Like, this is not the grams of, potentially magic mushrooms. This is the grams of the active ingredient. And in the mushrooms, it's something like 1 to 3% of the dry weight is the active ingredient. So you can go ahead and do the math. This is already huge quantities in just a liter volume. Like, you imagine an entire gram times 100 in a single liter to calculate like the biomass of dried mushrooms that you would be producing in 1 liter. And to be clear, please don't demonetize me. This is not a tutorial video. This is a video reviewing a scientific research article. I am a scientist. I have a PhD in bioengineering. We're just reviewing a research article. We're not telling you to go out and make any of these compounds in your basement or anything like that. So to be clear, in the last article, they used exogenous genes, exogenous meaning from another source. In this case, they used Psilocybe cubensis genes. In this paper, they're going to use various other genes from related fungi, so Psilocybe cubensis, sinensis, and then from different genuses as well, in order to potentially find some genes that in bacteria make higher quantities of these compounds. Now, this doesn't necessarily mean that these different species of mushrooms are automatically creating more of those compounds just because the genes work better in bacteria. Often when you're translating these genes between host organisms, they may function slightly differently. But going out into, the bioinformatics world and pulling different genes and trying them in your host organism is generally a good technique. So the conclusion of this paper is they found some new genes from other species that increased titers by nearly 50% to about 1.5 grams per liter of psilocybin, which is even more exciting than about gram per liter. And then finally, they end saying that this is sort of giving us a biosynthetic manufacturing route for psilocybin. And like a lot of metabolic engineering papers, they're often presented as, okay, we have this chemical synthesis route potentially for a lot of compounds that we want to make. We also have a natural synthesis route, so like harvesting from a plant, or in this case, a fungi. And then a lot of metabolic engineering papers will say, okay, well, both of these methods have their drawbacks. Extracting from natural resources uses a lot of land and water. And then chemical synthesis creates unnecessary byproducts or uses petroleum distillates or something like that. but a biosynthetic route has much fewer drawbacks, potentially, depending on the compound. So that's sort of the argument that's presented here. And sort of a little bit more motivation on making psilocybin specifically. So it's been part of, or it's currently part of 3 FDA clinical trials, which is really cool. And again, this section here is sort of talking about, traditional methods, chemical synthesis or whole organism farming have their downsides. But in 2017, the first group to publish the entire pathway, the biosynthetic pathway for psilocybin, pushed their work through. And this often happens or has been happening with THC and opioids and a bunch of other compounds and more compounds in the future. We first need to know what enzymes are actually making these compounds before we can make them in genetically modified organisms. And so that's what this is talking about here. So literally a year later, another group expressed these genes in Aspergillus nigulans, which is a filamentous fungi. It's a mold. And created some titers of psilocybin. They were only making 100 milligrams per liter, but that's still pretty cool. And then in 2020, the pathway was expressed in Saccharomyces cerevisiae. I have a video on that paper as well. But there were some issues here. They were making a lot of psilocin as well as psilocybin. And Dr. Jones had a really interesting take on this, producing these compounds in yeast instead of bacteria, because from my perspective, I would imagine taking genes from a fungi and putting them in another fungi, so magic mushrooms to yeast, would be more productive than switching kingdoms of life entirely from filamentous fungi like magic mushrooms into bacteria. But his point was that fungi, or yeast, since they're related to the magic mushrooms, they may have enzymes that actually degrade some of these tryptophan compounds. And so potentially expressing these same enzymes in bacteria could give us higher yields. And sure enough, that seems to be what's happening in the literature. Okay, but the downside of using bacteria is they are not good at expressing these P450 enzymes, which are these genes that are only found in eukaryotes, as far as I understand. And so for an E. coli, you have to feed the precursor this 4-hydroxyindole compound, which potentially is kind of costly and kind of difficult to get your hands on. But some people comment on these videos, oh no, like what if one of these genetically modified organisms gets in my body and starts infinitely making psilocybin or DMT or something, and I'm just tripping forever. That's probably not going to happen, because you need to feed this 4-hydroxyindole compound. First of all, it's probably not going to happen in general, but especially in this case, because you have to feed this precursor in order to make the downstream product. So using this approach where you have to feed a precursor rather than de novo, meaning from nothing, from glucose, may be potentially safer for, genetically modified organisms escaping into the environment. But again, that's a very low probability. These labs have really strict protocols of pasteurizing anything genetically modified before throwing it away. And there's other safety mechanisms in place as well. These organisms are pretty weak. We've highly expressed these genes that are very toxic to the cells, and so they're going to quickly get outcompeted by other organisms in the environment. They're very wimpy. Okay, so each species of psilocybin-containing mushrooms accumulates these latter N-methylated molecules in distinct ratios. So let's look at this pathway here to sort of clarify the thing that I was just talking about a second ago, as well as talking about this methylation step. So the pathway to make psilocybin from these commonly occurring compounds, we have 4-hydroxyindole, which is something that needs to be supplied to the bacteria. And we have serine, which is just an amino acid that the bacteria can synthesize on their own. And then we need this TRPB gene, which has very, very low activity in bacteria, so they just don't express it at all. And then we have 4-hydroxytryptophan. Psi-D is a gene from Psilocybe cubensis that converts it to 4-hydroxytryptamine. Then we have this Psi-K step, which converts it to norbaeocystin. And then now we're on the methylation steps that utilize Psi-M. So, um, you know, this Psi-M gene is going to be continuously methylating this amine group here with the nitrogen into psilocybin and then ureginosin. So we have norbaeocystin, which is a precursor, potentially an active molecule. Nobody really knows yet, so this is exciting. One methylation step creates baeocystin, which again, potentially an active molecule. And then adding another methyl group here makes psilocybin. One more methyl group makes ureginosin. And this pathway is really similar for DMT as well. So dimethyltryptamine, there's two methyl groups just like psilocybin. But again, potentially there's activity of these upstream and downstream compounds. And then we also have in this pathway the conversion of psilocybin to psilocin and vice versa, caused by this dephosphorylation and rephosphorylation right here. So psilocin is the actual active ingredient, and a similar pathway is going to be happening with ureginosin, baeocystin, as well as norbaeocystin, where this phosphate group will come off and likely create an active form of the molecule. But again, we don't really know how that works or the activity of these compounds. So very exciting new research. Reminds me a lot of the cannabinoid platform. You know, everybody knows THC. More people know CBD as well, but there's over 100 other compounds that are similarly structured that can be either chemically or enzymatically synthesized between other cannabinoids. And so we're slowly elucidating that same platform for tryptophan. So now we have this tryptophan platform. Everybody knows psilocybin, psilocin, and DMT, but there's also dozens, maybe hundreds of other related compounds that we're probably going to be synthesizing and testing in the next decade or two. So very cool stuff. So what this was talking about here is that different psychedelic mushrooms are going to be creating different ratios of these various methylation tryptophan compounds, which is cool. In this paper, they're sort of optimizing the different genes in the pathway, and they're doing that by testing genes from these different species. And so here are some of the acronyms. One to point out is the Psi-Q or SIQ, whatever you want to say, for Psilocybe cubensis. And then there's the Psi-C. These are great acronyms. PAN-C, and then GIMD for the different species. So we'll see these acronyms throughout the paper, so try to keep those in mind. And then here it's sort of saying the results at the end of the introduction, that the best psilocybin producing strain, which the strain was called GIMD30, which uses a Psi-M from the GIMD mushroom. How do we say that? Gymnopilus dilipis, something along those lines. We'll call it GIMD for now. But using the Psi-M from GIMD, they created the GIMD30 strain, and it's producing that about 1.5 grams per liter of psilocybin. And keep note, this is in a fed batch bioreactor. So meaning you have a bioreactor that's growing the organism and you're pumping glucose in over time. You may not be exactly continuously feeding in the glucose, but you might actually just be measuring the glucose, and if it runs out, then you add more. And this isn't, I wouldn't say this is cheating, but it's a way to increase titers, right? If you were growing in 2 milliliter culture tubes, or if you were growing in shake flasks, that shake around in the incubator, you're not able to continuously feed glucose, and you're not in optimal conditions like you would be in a bioreactor. And so potentially with those methods, you would be creating less. So just keep that in mind. This is already sort of scaled up. Because typically, if somebody reports that they can make 100 milligrams per liter in a 100 microliter culture, like in a well plate, then by the time you get the bioreactor, scale, you may be making grams per liter. So typically, not always, but typically when you scale up, at least from the lab scales, you're going to be making more and more and more. So this is sort of the top of the lab scale, what I would say. So then this Psi-M from psilocybe cyanescens were shown to accumulate higher titers of the intermediate biosystin. So this is also interesting that these different Psi-M molecules may have higher activity or lower activity on more or less methylated tryptamine compounds. Some people get mad in the comments when I don't go over the methods, but like these details are often sort of silly to talk about in a video, but I will briefly go over some of the details. But they're using two different strains of E. coli here. This DH5 alpha strain is commonly used to just produce plasmids, to produce these DNA cassettes. And then BL21 star is a common strain that's used for protein expression. So in this case, they're using that strain to produce the enzymes to make psilocybin, but they're doing it all within the cell. So BL21 star, common patented strain that produces high levels of proteins, enzymes of proteins. So if we express these psilocybe enzymes in this strain, then we should get really high quantities of these enzymes. And then the DE3 is a special addition, genetic addition to that strain. They're using this media that they're calling Andrew's Magic media for producing higher quantities of tryptamines. And then in some cases, they're saying they supplemented with serine and methionine. So we can see in this pathway that serine is needed. And then methionine is also needed. I think that was discovered in the previous paper. I forget exactly which step methionine is. Oh, right here. Methionine is needed here to produce this cofactor for Psi-M. So we need serine and methionine. So if we supplement it into the media, potentially we get higher titers, and I think that's what they found. Okay, so yeah, when they needed to create psilocin, they did it in situ, which means not in the cell, using a alkaline phosphatase psilocybin that they purified in-house. So they made this phosphatase, which is an enzyme to convert psilocybin to psilocin when they needed to. Okay, this gene sourcing and initial cloning, Uniprot is a website that has DNA sequences. And so you can go get DNA sequences there. In their supplemental materials, they have the DNA sequences, which are not illegal. Interesting point there. And they ordered these DNA sequences from a company called Azenta. They used to be called GeneWiz. And that's the same DNA synthesis and sequencing company that I used in grad school. They're fine. To give you a frame of reference, if you want to synthesize like a 1 or 2 kb gene, kilobases, so 1,000 to 2,000 base pairs, it's something like $200 to $500 in that price range. Some of these genes might be closer to 4 kb or 5 kb. But basically, each gene you want to synthesize is going to be less than about $1,000, which is just a crazy thought that you can just call up this company and say, hey, make this gene for me, and that gene costs about $1,000. There's, of course, additional steps where you need to clone it into a plasmid, which is basically a CD that you can then insert into the bacteria to get it to run. I'm saying it like it's code. It's basically code, right? Which is super cool. And then there's additional motifs that you might want to add. So it's not quite as simple as just like copying and pasting and clicking order and then shoving it in a bacteria. There's a lot more other little steps. And I think if you want to learn those kinds of things, there's a lot of videos on my channel that sort of introduce those concepts. Things like codon optimization, transformations into E. coli. I think I have videos for both of those on the channel if you're interested in learning more. The plasmid that they use, so when you want to run a program in an organism, you usually have like a plasmid backbone, which is like if you're using a CD or a floppy disk or whatever. They're different sort of systems for running the software in the organism. In this case, they're using a plasmid called PET M6H10 M cherry. So H10 is PET M6 is a plasmid. Lowercase p means plasmid. H10 is a promoter, and then M cherry is a protein a protein that's red. So when this is expressed, the E. coli will be red. So all they did in this paper is they took this existing plasmid that's out there that anybody can order and replaced M. cherry with the Psilocybe cubensis or other fungi genes. Super cool stuff. And then they're talking about their libraries here a little bit, and basically what they're doing, and we'll talk about this more in a second, is they're trying out different promoters and different enzyme combinations in order to make higher titers. They have different promoter sequences that they're working with here. So T7 is a high strength promoter, so it's going to turn the gene on way up. It's going to make a lot of the gene. And then there's lower strength promoters. Some of the promoters are inducible, so when you add a chemical, then the gene is turned on, which can be beneficial. We'll talk about that more in a moment. A few other pieces. So their fermentation conditions here, when they're doing their library screening, when they're trying out their different cultures, they're doing it in 2 milliliter cultures in 48 well plates. So the well plates are about this big, right? And it's split into 48 cells. And each cell has 2 milliliters of culture. They're grown at 37 Celsius. And then they're talking about their bioreactor conditions. They're probably using a 2-liter bioreactor, but with a 1.5-liter fermentation volume. So they're using 1.5 liters of media in a 2-liter bioreactor. And it was grown at 37, which is very typical for E. coli. And then they're saying their supplementation with the 4-hydroxyindole was supplemented at 150 milligrams per liter in the bioreactor, and then they varied the feed rate. So not only are they supplementing glucose throughout time, but they're also supplementing the 4-hydroxyindole, which is the precursor. And they must have added a lot or found out that 4-hydroxyindole is at least partially toxic to the cells, because if they're making 1.5 grams per liter of psilocybin, then they need to be adding, well, you would have to do the molar calculations, but on the order of grams per liter of 4-hydroxyindole. So they're probably having to add something like 2 grams per liter total of 4-hydroxyindole, and that gram per liter amount may be toxic to the cells. So that's something important here, that, if this 4-hydroxyindole is toxic or it's being degraded too quickly, they must have found that you need to slowly feed it in over time in order to get the highest titers. So another detail here is they're evaluating these metabolites in triplicate. So for each of the experiments they're doing, they're measuring the production 3 different times, which is pretty solid. For their analytical methods, they're using HPLC, high performance liquid chromatography, with a diode array detector. These are really cool, and a refractive index detector. So RIDs are typically used to quantify sugars, which I think is what they used here. And then diode array detectors actually measure. So the HPLC is basically a black box that you send a chemical through, and you send a sample through and it separates it into individual chemicals. And on the other side of the HPLC, you need detectors. So again, the RID is best for detecting sugars coming off of the column. And then the diode array is often called a photodiode array, or PDA, or in this case, it's called a DAD. It can measure various wavelengths. So maybe psilocybin happens to absorb, 700 nanometer light really well. Then you need to see at 700 nanometers in order to literally see the compound coming off of the column. Anyway, analytical methods are really cool. And then they also used LCMS, so liquid chromatography coupled with mass spectrometry. Mass spectrometry takes the chemicals coming off of the HPLC, ionizes them, and sends them through a really strong electromagnetic field to split them up into their individual components. And it gives a chemical fingerprint, so you can actually identify which chemical is coming off anywhere in the world. You could be anywhere in the world, and as long as you know the chemical fingerprint of the molecule that you want, you don't need a standard. You just send it through and you can see the fingerprint and you know you're making that chemical. I have a really good video on my channel that I just made. It's like a whiteboard style video for some analytical techniques if you're interested in that. So they use 2 microliters of volume from the supernatant. So they're taking like a milliliter of the culture, spinning it down, taking the supernatant, 2 microliters of the supernatant, and then injecting it into the liquid chromatograph. Okay, it turns out that 280 nanometers is the wavelength that you need to measure these tryptamines, so that's important. And these details are the column. So this is a C18 column, so it's a non-polar column, from what I understand. And they're using the mobile phase of water and acetonitrile. So in these liquid chromatographs, you have a mobile phase and a stationary phase. The mobile phase is going to be, either polar or non-polar or a mix of the two liquids going through the stationary phase, which is just a tube packed with tiny particles that are either polar or non-polar or have different material properties, and passing the mobile phase through the stationary phase with your sample going through as well ends up separating your compounds. Okay, so that's everything here. So for measuring tryptamines, you need to be using this C18 column with water and acetonitrile as the mobile phase. And this water and acetonitrile are varied over time. You can see the details there. And then for sugars, they're using an HPX 87H column. And again, they're using that refractive index detector for the sugars. And their mobile phase in that case is just sulfuric acid and water. So dilute sulfuric acid and water. All right, we have survived the materials and methods section. Congratulations. Okay, confirmation of Psi-D, Psi-K, and Psi-M activity. Additionally, due to limited commercial availability of select intermediates and or membrane transport limitations surrounding direct supplementation of phosphorylated immediate precursors to the culture media, Psi-K and Psi-M variants were tested in combination with previously verified genes from Psilocybe cubensis. So Psi-K and Psi-M, let's go back to this here. Psi-K and Psi-M were tested, but they couldn't get this 4-hydroxytryptamine. Either it was expensive to order or impossible to order, or when they added it to the culture media, it wasn't taken up by the cells. And so in order to test Psi-K and Psi-M, they had to also express Psi-D and then feed 4-hydroxy indole, right? So moving on. That's important. And they're testing not only the different genes, but also different promoter sequences. So they have an H10 mutant T7 promoter. So that's one of the promoters that they're using here. So these constructs were expressed in basic operon form under the control of H10 mutant T7 promoter. Okay, sorry. First, they're starting with just that promoter. Let's look at this figure here. First of all, highlighting here, this XX4 superscript 4 denotes one of 4 possible sequences. So for Psi-D and Psi-K and Psi-M, there's 4 potential genes from the 4 different organisms that they're working with. They're taking this construct, which uses a C4 promoter, and then this construct that uses an H10 promoter. Let me see if I'm missing something. I'm not sure what the blank promoter gene means, or what the blank promoter means. Maybe that's no promoter as a control or something. But anyway, they're shuffling these together and combining these with different combinations. So then here you could have one promoter promoting two genes, either psi D first or psi K first. Sorry, psi D is first here, psi K is second, and these are two different promoters. And then you can also have all of the genes driven by the same promoter. So basically this is a schematic that's showing how different ways that we can combine the genes and different promoters to optimize their activity. So moving on here, they're saying they created a potential library size of 64 mutants. So 4 to the power of 3. So H10 mutant T7, medium strength C4. And then whether or not the N-terminal T7 epitope tag was present on the construct. So this is just a tag on the gene. So basically they have this library of different genes in different orders with different promoters, and they're trying to find which one works the best. So in this case, in this figure here, we're looking at psi D different with different genes. So this orange line is the gene from this pant seat, and then we can recognize psi Q or Psilocybe cubensis in this sort of gray line here. So this is expressing a psi D from Psilocybe cubensis, and then this peak here is for, let's see, for 4-hydroxytryptamine. So it's that first compound in the pathway. So 4-hydroxytryptamine. So we're testing psi D, we're testing this in panel A, and so we're seeing which gene makes the highest quantity there, right? So you can see that in the green, psi C doesn't really make any, and the control also doesn't really make any, so that's important to know. And then now we're looking at the next compound, which is norbeocystin in the pathway. And you can see this green, so psi-C, Psilocybe sinensis is making the most of this compound. And then finally having the entire cassette there to make, let's double check this. For psilocybin, yep. The Pansy organism is making the highest amount of psilocybin. And this is all LC-MS data, I believe. Yep, mass spectrometry data. And that's why we have these MZ values, which is mass over charge. Okay, and then we're looking at, in this figure, we're looking at different library or different mutants pulled from the library. So you take, you have a potential of making 64 mutants. You have this DNA library that you then transform into a batch of cells. You put them all on Petri dishes, and hopefully you're getting like 10 times or 5 times at the very least library coverage. So maybe you're getting 600 colonies. And then you can pick individual colonies and grow them in liquid culture and then measure how much of the psilocybin they're producing. So here we're looking at psilocybin production and it's normalized. So potentially in each batch, they had triplicate of this control strain. And they say somewhere what the control strain is. I think it's a previous strain that makes high titers of psilocybin. So then by having that control strain on every plate, you just make sure that the conditions in every run were the same. And so they just normalize the psilocybin production here. So this top strain here, you're probably most curious about, is making about 10% more psilocybin than the control strain. It's using the C4 promoter. And then let's see if it says what 235 is. So 235 uses Psilocybe cubensis genes for all 3 genes. So in this case, the winner is the Psilocybe cubensis genes for all of them, if you're trying to make psilocybin. And then another, so the H10 promoter was also good, so that's 360 and 296. So the C4 and H10 promoters were both good for all of the Psilocybe cubensis genes. So C4 is in pink, control production. Okay, so the control is Silo 16, which is, I believe, the best producing strain from the previous paper. So we're already getting higher psilocybin production in these 3 different strains. 92 variants were screened per library. So if we have 64 potential mutants, we're screening 92 variants. We have like 1.5 times coverage of the library. So potentially we're not seeing every mutant in the library, but that's okay. We don't need to see all of them. We just can't say that these are the best ones in the library because we may have missed some of the mutants. Because they say here, 74% of the clones were found. Oh, never mind. That's not what this is saying. What this is saying is 74% of the clones were found to have no measurable psilocybin production. So 3/4 of what they tested didn't have any psilocybin production. Oh, and I got this wrong. It's 92 variants per library, and I think there were like 4 libraries of 16 or something like that. Anyway, it's higher coverage than 1.5. They tested most of the strains. And then they say here, strains with the asterisk were selected for further evaluation. So of course, these top 2, this one, this one, this one, were selected for further evaluation, and some of these as well. So this one here is with the T7 tag. So this A panel is with the T7 promoter, which I believe is an inducible promoter. And then this one is without the T7 tag. And you can see this produces way more psilocybin, which is cool. All right, so let's make sure we're not missing anything. Evaluation of the variants, development of P-Nor, which produced 1.58 grams per liter. Okay, this is talking about a different strain that produced high quantities of norbaeocystin from a previous paper that you could check out. Right, okay, so this was saying here that norbaeocystin is probably not the limiting, production of norbaeocystin is probably not the limiting step. It's Psi-M that's the limiting step. And so they're saying now they're going to test these methyltransferase variants, which is Psi-M is a methyltransferase. They tried doing this co-culture screening where they added 2 strains, one with Psi-K, or one that produced, let's look at the precursors. So just to be clear, one strain that produced high quantities of norbaeocystin, and then one strain that produced a variant of Psi-M, and they mixed that in a single culture to sort of find high producers of Psi-M. And they said that this didn't work very well. And I think it's really cool that they reported something that didn't work well, right? It's good to see negative results. The goal was to provide norbaeocystin produced by the previously optimized P-Nor strain. Anyway, in co-culture, which would then be taken up by the methyltransferase containing strain. And then what they found was, okay, so they skewed inoculation ratios. So, however, the absence of psilocybin or baeocystin production, data not shown, suggests a mass transport limitation. So what's happening in this cult culture, what they were hoping would happen is that they have one strain producing norbaeocystin, it goes into the media, and then it goes into the mutant Psi-M strain, and then it's converted to psilocybin. But what they found was there's probably some limitation either of norbaeocystin going into the media, which is unlikely, or norbaeocystin going into the Psi-M E. coli cell, which is the most likely option. So that's really interesting. And what this suggests is that you need to produce the compound in one organism. Everything needs to happen in that one cell. And maybe it may even suggest that you want to keep all those chemicals in the cell until psilocybin is produced. And that's another interesting thing here is that we may be, you can't say for sure that these different genes are purely pushing the chemical reaction one way because of their efficiency. Like maybe one enzyme has, has a longer life in the cell or something like that, and then one enzyme is, very quickly making a lot of the chemical and then it's being degraded quickly or something. And maybe one of those methods is beneficial in the cell. And so maybe in the cell, what we're optimizing for actually is rapid conversion and not necessarily enzyme integrity or, bias towards how many methyl groups it adds or something like that, right? So, yeah, so again, they're saying, like I just said, that transport out of the cell is not impeded. Right. 4 independent transcriptionally varied libraries were cloned in operon configuration, each with 7 possible promoters. So now they're testing 4 genes with 7 possible promoters in a strain that already produces norbiocystin. So now they're doing it all in one cell. They're varying the Psi-M gene from these different organisms, again, with these 7 different promoters. Xyl A, and GAP are constitutive promoters, meaning that they're always on. And I might have been wrong earlier about this, but 5 of the promoters, the other 5 promoters, are IPTG inducible. So IPTG, forget exactly what it stands for, but it's just a chemical that induces these genes. It turns on these genes when you add them. So theoretical library size of 7 constructs for each 4 pathways. And then in their screen, they screened over 5 times the theoretical library size to make sure that they got all of the strains, or at least they got most of them. And in this method, they selected one high psilocybin producing strain, PsiQ1, that was making 338 milligrams per liter. And again, this is in just a 2 milliliter volume. So when we scale up to bioreactors, it's going to be more. They also went through with a lower producing strain just to see what would happen with that one. Pathways containing the GIMD Psi-M gene presented many strains of interest, with the top 3 randomly selected colonies producing an average of 500 milligrams per liter of psilocybin, which is awesome. GIMD configurations resulted in specific compositional enhancements in any of the other methylated products of interest. Okay, so that's that section. This is showing the, comparing Psi-M between the different organisms. And it's sort of showing the profile, right? So you imagine that, in cannabinoids, we talk about the different cannabinoids that can be present in any strain. People are starting to talk about entourage effects in psilocybin mushrooms. So this combination of, biosystin, norbiocystin, psilocybin, psilocin, the exact composition may have different effects. And so we see this in bacteria as well when we express these genes in bacteria, that in this Psilocybe sinensis strain, this particular promoter gene combination is producing 100 milligrams per liter of biosystin, but in Psilocybe cubensis, this particular combination is only producing 50 milligrams of biosystin, but the same amount of psilocybin. So potentially this may be interesting. Maybe you want some a Psi-M that produces equal amounts of psilocybin and biosystin, or no psilocybin at all and only biosystin. So this is some cool stuff to know. And then this is showing some sort of details here of, so this is the Xyl A promoter on GIMD 30, and it's producing really high quantities of psilocybin and relatively low quantities of biosystin. And then we have a few other comparisons here. All right. So now they're looking at producing in bioreactors. So to determine full potential of these strains, lead strains were selected based on potential for either psilocybin, so GIMD30 and PANC10, or biosystin, so PSIC30 or GIMD22 production, according to their performance in the small-scale screening. So they used Andrew's Magic Media supplemented with 5 grams per liter of methionine and serine. And then Biosystem production strains were grown without the supplemental methionine because, again, the methionine is only needed for cofactor production in the Psi-M step. So if you're producing, or I guess this was previously found in another paper, if you're producing Biosystem and you add, or if you're trying to produce Biosystem, you should not add methionine so that more methylation steps don't occur. That makes sense. Okay, restricting methionine supplementation increases overall titers of norbiocystin, like I was saying, and biocystin. So 0 milligrams per liter to 178 and 162 to 348. So it's like doubling the biocystin production, and it's actually giving some visible amounts of norbiocystin. And correspondingly decreases titers of psilocybin. So that's good to know. So, it's cool, and this is another advantage of biosynthesis is we can vary the media composition in order to change what compounds we're producing. So moving on, these are the bioreactor or one of the bioreactor experiments. This is for GMD30, which potentially is the best strain here. And you can see psilocybin titers get up to that advertised nearly 1.5 grams per liter level. But you can also see the biocystin levels are down here, 4-hydroxyindole, 4-hydroxytryptamine, 4-hydroxytryptamine, maybe that's tryptophan. But 4-hydroxyindole is the precursor that's added. So you can see there's start out with 150 milligrams per liter, like I said in the beginning of this review. It drops down. They probably collected their HPLC data and they realized it was down, and so they started adding more. And you can see that this goes up because they're adding it to the media. And same with the glucose here. The glucose is consumed pretty quickly, and then they add some glucose to keep the culture going. And this seems, oh, right, because glucose is over here. I was like, 800 milligrams per liter of glucose is not very much. They're starting with about 20 grams per liter of glucose, which honestly still is kind of low. But then they're controlling it. And then they're also measuring the OD600, which is basically the density of the culture, the optical density at 600 nanometers. So how much 600 nanometer light goes through the culture, which is a proxy of how many cells are in the culture. And then this graph is just showing the dissolved oxygen is kept at above 25%. So they're starting at like 80-90% oxygen, which is really high. DO percentage, yep. So they're actually sparging oxygen and air probably into the bioreactor. Agitation, so this is the speed of the impeller, that helps increase the dissolved oxygen as well. So that's why that's changing over time. You can see once the dissolved oxygen drops, the impeller kicks on so that more air gets dissolved into the culture, more oxygen gets dissolved into the culture. You can see the pH is kept constant and the temperature is kept pretty constant at 37. So that's the bioreactor. So previously, okay, so that's basically all the data here. And now they're talking about cost, which is interesting. So previously, the Silo 16 bioreactor data, they estimated the cost per gram of psilocybin at a reasonable scale, maybe like hundreds of liters, is about $9 per gram of psilocybin, which is so cheap, like so cheap. That's like $1 per dose or less. Crazy cheap to produce. And then for this new GMD30 strain that's producing even higher titers of psilocybin, it's now closer to 80 cents per gram. Or sorry, it's an 80-cent reduction. So now it's closer to $80 per gram. And part of this cost reduction is not needing IPTG to increase titers. So IPTG is that inducer that you add to turn on the genes. So they developed a strain that doesn't need IPTG induction. So maybe we can go back and look here and clarify that, right? Okay, so here's GMD30. It uses ZAL-A, which is a constitutive promoter like we just talked about. So you don't need IPTG. And so that's a big chunk of the cost reduction there, which is cool to see. So we conclude that Psi-M is capable of ureginosin formation. However, this activity is markedly lower than its activity towards other norbeocystin substrates and therefore is only detectable under optimal conditions. So I guess that was a question in the literature, whether you can make aeruginosin. So here's aeruginosin. Psi-M is going to have activity on this amine group, whether or not it's methylated. But I guess they're saying that typically this Psi-M activity is really, really low the more methyl groups you add. And so actually producing this aeruginosin is hard. But they did some experiments in this paper to show that you can make aeruginosin with just Psi-M. So that was sort of another cherry on top. And there's this theme in metabolic engineering papers and maybe in other fields as well, where you sort of have like a good, neat story of like, we were producing low titers of psilocybin, and in this paper, we're screening a bunch of different enzymes and promoters to make even more psilocybin. And look, we did it. We made more psilocybin, and that I feel like has a nice bow on top of like, this is a full story. But I think there's been this push, and I don't know if this is a recent thing, to sort of add a little cherry on top at the end that isn't directly related to the story, but sort of flexes your expertise in some ways, or flexes a new technology that you have access to. And so in this case, what they added, proteomic analysis reveals energy limitations for continued high psilocybin production in the GEM-D30 strain. So they're looking at proteomics, which just feels a little out of, I mean, it's important, right? But this is sort of like, it feels sort of tacked on to the end. But again, this is very common in, even like Nature papers, they do this kind of stuff. So anyway, if you have comments on this, leave them in the comments below. But I've always felt it's a weird thing that just happens. Not dashing, this particular lab group, like everybody is doing it, and I just don't fully understand it. So here they're saying the key biosynthetic enzymes are expressed at levels comparable to core metabolic enzymes, enabling a balance between metabolic burden of pathway overexpression and robust biosynthetic activity. So this is another thing that can be optimized in metabolic engineering is you can way overproduce enzymes if you want to, but it will make the cells really unhealthy. And so if you don't have cells growing, then you don't have the production of those enzymes. So there's sort of a balance between cell growth and production of your enzymes to make your product. And so they're saying basically they had a good balance in those things. And then this here is called a volcano plot, and that's all I know about this figure. So really cool paper. If you have questions, please leave them in the comments below. If you have answers, please leave them in the comments below. If there is a research article that you want me to review next, let me know below. I am open to consultation work if that's something that you're interested in. Producing these compounds would not be very hard. I'll say that. So open to consultation. You can find my email in the description below, and the Instagram for the DIY Biotech YouTube channel. Thank you so much as always for watching. Make sure you like and subscribe to the video. I'll see you in the next one. Bye.